Hello everyone, welcome to Lab Science Tutorials by Archana Agustin. In today's video, we are going to discuss platelet in more detail. Especially, we will cover two headings, the production of platelet and their ultrastructure. First, let's discuss the production of platelet. This process is called thrombopoiesis. Before we get into thrombopoiesis, let's revise two key points about hematopoiesis. Hematopoiesis means the production of blood cells that include RBC, WBC, and platelet. In adult, hematopoiesis occurs mainly in the bone marrow of flat bones like sternum, skull, pelvis, sacrum, and vertebrae. It changes with age. In fetal life, it starts in the yolk sac, then shifts to the liver, and finally to the bone marrow. All blood cells comes from one mother cell called pluripotent hematopoietic stem cell. The stem cell divides into two lineages, myeloid lineage and lymphoid lineage. Platelet comes from the myeloid lineage. The myeloid progenitor gives rise to granulocyte, which are eosinophil, basophil, neutrophil, erythrocyte, monocyte, and megakaryocyte. And the megakaryocytes are what produced platelet. However, common lymphoid progenitor cell or the lymphoid lineage cell will give rise to T, B and B lymphocyte along with natural killer cells. Now let's talk about thrombopoiesis, that means the production of platelet. Platelet are formed by the fragmentation of cytoplasm of megakaryocyte. This happens inside the bone marrow sinusoids. The whole process is controlled by a hormone called thrombopoietin or TPO. TPO is mainly made by the liver and it will increase when the platelet count in blood falls. Here is the diagrammatic representation of platelet production. The earliest precursor is megakaryoblast, size from 20 to 30 micrometer. It has a large nucleus with 1 to 2 nucleoli. The megakaryoblast undergoes endomitosis. That means the DNA and the nucleus keep replicating, but the cell doesn't divide. So one nucleus becomes 2, 2 becomes 4, 4 becomes 8, and that is known as endo-reduplication. The next stage is promegakaryocyte. Size increases, but it still doesn't have granules in the cytoplasm. Then comes megakaryocyte. This is the giant cell and its size ranges from 30 to 90 micrometer. It has 4 to 16 nuclear lobes and the cytoplasm has numerous granules, alpha granules and dense granule. Finally, the megakaryocyte undergoes cytoplasmic fragmentation. The cytoplasm breaks off into tiny pieces and each piece becomes a platelet. Platelets are disc-shaped cells present in the whole blood. The normal concentration is about 1.5 to 4 lakh per microliter. There are non-nucleated cell fragments and their main function is hemostasis. Now, what do you mean by hemostasis? That means stopping bleeding and forming a clot. For platelet estimation in the lab, we use Leishman stain. We can use a Wright stain, and also for the estimation of platelet count, we can either do manual counting in Nuba counting chamber, or we can use automated hematology analyzer. If you see the picture here, we can see there are numerous microtubules present just beneath the cell membrane. These microtubules will maintain the integrity and shape of the platelet. Without them, platelet would lose their shape. In the cytoplasm, you can see contractile protein, which are actin, myosin, and thrombostatin. These protein help in the clot retraction. They pull the wound edges together. And there is also mitochondria, endoplasmic reticulum, and Golgi complex present in the cytoplasm. Then the important feature, that is the granules. So we have two type of granules in the platelet. One is alpha granule and another one is dense granule. Alpha granules are larger in number and size. They store basically fibrinogen, von Willebrand factor, protein derived growth factors, etc. Then there is dense granule, which are smaller, and they store ADP, ATP, calcium, etc. The cell membrane of the platelet has glycoproteins that act as receptor. This receptor bind to chemicals like collagen, thrombin, ADP, or anything that can activate platelet. Now let's see the composition of platelet granules. Remember, platelet have two type of granules, alpha and dense granule. Alpha granules stores protein derived substance. The composition of alpha granules are PDGF, that is platelet derived growth factor. This factor will stimulate the endothelial cells and fibroblast for wound healing. Then there is coagulation factor present, which is factor 1 and factor 5. Factor 1 and 5 are basically synthesized from the liver. Then von Willebrand factor, this is an important for platelet adhesion. The von Willebrand factor is synthesized by the megakaryocyte, and this is stored inside the alpha granules. Finally, let's talk about the composition of dense granules. Dense granule contain non-protein substance, non-protein derived factors such as ADP, ATP, serotonin, and calcium ion. ADP and ATP will help platelet to bind to the neighboring platelet, which that helps in the aggregation of platelet. Serotonin is a vasoconstrictor. It will help vasoconstriction. And then calcium ion has an important key role in the coagulation. So this is all about platelet and its ultrastructure. So if you like this video, please do like, share, and subscribe to Lab Science Tutorials. In our next video, we will discuss more MCQ and practice questions from this session. So stay tuned. Thank you so much.